Good morning, everyone. First of all, I would like to thank the scientific committee and the partners for allowing me to present my results. So my topic is dealing with coagulation of proteins, a molecular aspect of skin aging. So proteins over their life, they are exposed to post-translational modifications. So we can call them young at the beginning and old at the end of their life. And so first, they are subjected to enzymatic modifications, which are reversible and necessary for their structure and their functions. We can, for example, cite methylation, phosphorylation, acetylation. And later, they are exposed to non-enzymatic modifications, so after minutes, hours, even years. And these modifications are irreversible. And they are responsible for the loss of their structure and their function. Among them, we can cite oxidation due to reactive oxygen species, very well known, glycation due to reducing sugar, and also caramelization. So I'm going to focus on these two modifications. Caramelization is due to the binding of the isocyanic acid to free amino groups of proteins. And when this binding occurs on the epsilon NH2 of the lysine residues, it generates the caramelated lysine, also called homocitrulline. So all of these products are called CDPs for caramelization-derived products. This molecule, isocyanic acid, mainly comes from the spontaneous dissociation of urea in the blood. But it might come from, thiocyanate through the reaction with the enzyme myeloperoxidase or from atmosphere. But these two sources are very minor. Glycation is not a one-step process, but a successive process involving many reactions. So briefly, a reducing sugar will bind on the amino groups of proteins, generating a Schiff base and then an animatory products. And in later steps, these products are exposed to further reactions like oxidations, cleavages, crosslinks, leading to the formation of a wide group of AGEs, advanced glycation end products. So many of them are very well known, but I can cite the CML, the carboxymethyl-lysine, or the pantozidine. These modifications have many consequences. So first, they are responsible for the alteration of the structure of proteins. This is the case, for example, of type 1 collagen. This modification alters the fibers. So you can see here on the top, the native type 1 collagen, and on the bottom, the caramelated type 1 collagen. So we can clearly see that the network is very disorganized. And consequently, these modifications generate an increase of the stiffness and an increase of the mechanical resistance of the fibers. They are also responsible of inappropriate cell responses. For example, there is an increase of the inflammatory activity of human cells, an increase of the MMP's secretion, but also a decrease of the senescence of the fibroblasts, the decrease of their proliferation, an increase of their apoptosis, or also a decrease of the matrix synthesis, matrix proteins. So glycation has been widely investigated over the last decades, and especially because 20 years ago, even more, glycation has been described as aging process because the CML and the pathogen increased over time in skin or skin type 1 collagen. But about cumulation, much less data are existing. So that's why with my team, we suggested to study cumulation over skin aging. So for this purpose, we extracted skin from 3 different mammalian species with 3 different lifespan. So around 75 years for human, 20 years for bovine, and 2 years for mice. We extracted skin samples and we divided these samples in 3 sub-samples. So the first one is the total extract. The second one was submitted to an extraction and the purification of type 1 collagen. And the third one was subjected to the extraction and the purification of elastin. And from there, we quantified the homocitrullin, so the caramelated product, and the CML, the glycated product, to compare these 2 modifications by mass spectrometry. And we also evaluate the cumulation by immunofluorescence. So first, let's see the results from the skin extract. So in plain color, you can see, in plain color, you can see that there is an increase of homocitrullin concentration, so in all studied species, around by 11-fold in mice and bovine, and 10-fold in human. And in squared color, here, you can see an increase also of the CML, but in a less extent, so by 2-fold in mice and bovines, and 6-fold in humans. So we can say that cumulation is a physiological process, and that the homocitrullin formation is more important than the CML formation. But we cannot say that carbamylation is more important than glycation, because we observe just 2 products. But at least we can say that carbamylation is as important as glycation. Then we pulled this 3 graphs into 1 graph to observe the accumulation speed of CDPs. So if I zoom on the first years, you can see in gray that the linear regression is steeper in mice than bovine and human. And to clearly see that, I spotted in 1 graph the coefficient of the equation according to the mean life expectancy. And here we can see that there is a correlation between the life expectancy and the accumulation speed of CDPs. So in other words, we can say that longer the lifespan, slower the accumulation, and inversely, shorter the lifespan, faster the accumulation. We observed exactly the same trend for CML as previously described. So to further study the carbamylation in skin, we performed an immunofluorescence study in 3 different skin sections. So a subject being 20 years old, another one's 36 years old, and then an older one, 80 years old. So you can see the epidermis here and the dermis for each section, and in green, the homocitrullin labeling. So in the young subject, we can barely see the green signal, whereas in the older subjects, we can clearly see an intense signal. And this signal is very intense in the reticular dermis, not in the epidermis, not in the papillary dermis, but in the reticular dermis. And this dermis is very rich in collagen, and elastin, so two matrix proteins which have a long half-life, so around 20 years old for collagen and 70 years for elastin. So that's why we purified the type 1 collagen in skin from the three species as previously, and we observed similar results than observed with the total extracts. So meaning that there is an increase of homocitrulline in this species and a significant increase. And this increase was about 5-fold in mice, 11 in bovines, and 3-fold in humans. And as previously, we can observe also an increase of the CML, but in a less extent. So type 1 collagen is progressively accumulated with age and accumulates in the skin. And as previously observed for the whole extract, there is a correlation between the accumulation speed of caramelated protein and the mean life expectancy. Then we collaborate with Christian Schmelzer from Germany to perform a proteomic analysis by spectrometry on the skin type 1 collagen samples. So he did this analysis on 4 samples, and these samples were extracted from subjects being older than 77 years old. And we observed that per person and per molecule of type 1 collagen, there are 31% of total lysine residue which were caramelated, 20% were glycated, and 14% were either caramelated or glycated. And previous data showed that for lysine residues, so around 40% of total lysine, were enough to impair the conformation of the fibers and also to decrease the thermal stability and the fibrillogenesis of type 1 collagen. So altogether, we can speculate that caramelation is likely involved in the skin rigidity because of the disorganization of the type 1 collagen fibers. For the elastin, we got the result just for human. Unfortunately, I don't have the results in mice and bovines due to protocol limitations. So we clearly see here again that homocitrulline increased over time in human as well as the CML. So again, caramelation is likely involved in, or probably, this have to be addressed, involved in the elasticity loss of elastin fibers. So from this data, we were wondering if the caramelation can be prevented by the organism. So as preliminary results, we used a mice model. So the mice were fed 3 weeks with cyanate, so supplemented water for 3 weeks, and then we replaced the water by regular water for 3, 6, or 9 additional weeks. And as a control, some mice were fed just with regular water for 12 weeks. So first, we can see that when the mice were fed with cyanate, there is an increase of the homocitrulline concentration in plasma, skin, and type 1 collagen, as expected. But then, when we replace the cyanate water by regular water, there is a decrease of homocitrulline concentration. So almost, this decrease was almost complete in plasma, and that makes sense because The plasma proteins have a short half-life, so they renewed very quickly, and that explained this huge decrease. However, in skin and type 1 collagen, this decrease is weaker, so around 70% in skin and 45% in type 1 collagen. And also this makes sense because type 1 collagen has a long half-life. But there is a decrease just within 9 weeks. So we can speculate that some system processes are existing in the organism to prevent carbamylation. So we can speculate for the intracellular CDPs that they are degradative system by proteomic system. For example, proteasomes or autophagy. For the extracellular CDPs like type 1 collagen, we can hypothesize that they are subjected to proteinases to degrade them, especially because it has been described in our lab previously in vitro that carbamylated type 1 collagen is more sensitive to gelatinases like MMP2 and MMT9. And finally, we can speculate that some repairing systems are existing in the organism. So, bottom line, carbamylation is a physiological process and a hallmark of skin aging. It's a major non-enzymatic post-translational modification as relevant as glycation. It impacts particularly matrix proteins, which have a long half-life, like collagen and elastin, is responsible for the accumulation of this carbamylated product in the skin, and they can be eliminated from the organism. Of course, these preventing systems are puzzling questions that have to be addressed in further experiments. I would like to thank my team from Reims and also the team in Germany for having collaborated with us. And I would like to thank you, and I will be glad to answer to your question. Thank you.
Thank you very much for this excellent presentation. First question, perhaps, are there existing pathology who has to do with carbamylation?
Yeah, I didn't talk about pathology, but it's true that carbamylation was investigated first during chronic renal failure because there is a hyperuremia, so that promotes carbamylation. So that's why carbamylation was completely unknown during aging.
Thank you. Is there questions in the room?
My question is related to your answer. If there is increase in carbamylation with aging, is it because there is an accumulation of urea during aging?
So urea is stable. I mean, if we don't have any pathology, kidney pathology, I mean, the urea is stable, kind of stable. So definitely is probably because there is accumulation of long life carbamylated proteins. Because we quantified the homocysteine in plasma. For example, in mice, and there is no increase. And as I said, it's logical because the plasma proteins are are quickly renewed. So the accumulation is explained just by the carbamylation of long half-life proteins.
Question?
Thank you for this exciting presentation. The CML, the carboxymethyllysine modifications are, in the human skin at least, somehow connected to UV sun exposure. Is there something similar? Did you check for body site for your collagen samples, or is there
Yeah.
any connection to
So
exposure?
we had the same question, but we kind of knew the answer because carbamylation is not due to any oxidation reaction. But yes, we extracted skin from the belly, from the back, the forearms, the leg, and we didn't see any difference according to these different sites of the body. And as I said, it's not related to oxidation, but to urea. So, yeah.
Marek.
Okay, thank you for your talk. So I have two questions. The first one is, so do you know if this kind of mechanism could be involved in carbamylation of neuroproteins in the skin or in any kind of tissue?
If there are other tissue that...
You focus on collagen, for example. So
yeah.
Could you imagine something that could be relevant to translate this kind of approach in other type of neuroproteins that could be involved also in peripheral tissue, like alpha-synuclein for Parkinson's disease and beta-amyloid for Alzheimer's disease?
So I don't think that any studies has been done on this kind of pathology. However, as I said to someone in here, I extracted skin from many subjects from the Forensic Institute, and I remember that one of them was a little girl. She had epilepsy, and I remember that the concentration of homocysteine was very, very high as chronic kidney failure patients. So I don't know why. I just removed this data from my experiment, but it was very interesting, and I don't know the answer, but I think there is a link, I don't know, with some neuropathology. So, it was very, very interesting. So, but so far, no, carbamylation is largely investigated in chronic failure.
Okay, thank you. I see that there is, in the fields of your global collaboration, some people coming from a pharmacological institute. Which kind of pharmacological approach could you have on these fields for the future?
For the future, so different approaches can be done. So either a competition approach, so we can, for example, take amino acid to compete with our own proteins. So I think it's on the investigation right now. Another approach will be to prevent the first step of the reaction. So we can, for example, chelate the isocyanic acids and see if there is a prevention of carbamylation or not. To screen? This is, we don't know yet. So it will be interesting to study that.
Question from Marek.
Well, I don't know whether this is a question or just a comment, but it's spectacular to see that the process, which is supposed to be a chemical process, is tightly related to the lifespan of the animal or human being. It must be a kind of dynamics behind this, which is not related to the chemical interactions in the given organ, but as you say, some systemic probably changes that influence these peripheral modifications.
Yeah.
how to approach this?
Yeah, we were wondering the same question because it's just a spontaneous reaction, right? So first in mice, in the uremia in mice is a little bit higher than in human, maybe explaining that this accumulation is faster in mice. And then we can speculate that in human, because the accumulation is slower and we have a longer lifespan, so we can speculate that our organism has more anti-carbamylated system, like antioxidants that we have, or anti-glycation, system. So we can speculate that we have the same system for carbamylation. And I mean, we clearly see that for the type 1 collagen or the skin, for example. So, yeah. Mm-hmm.
Okay. Thank you very much. There is a good question. We have to call. Thank you very much, Mrs. Goris. So I call now. I call now Mrs. Mernaz Shamal Nassab to speak about salicylic acid derivative who can extend the lifespan of C. elegans. Mrs. Shamal Nassab is biologic researcher from University of Warwick now.
Hello everybody, this is Mernaz, and first of all I would like to thank the organizer for inviting me to be here to discuss with you the results that I have done at École Normale Supérieure de Lyon in Dr. Aguilano's lab in collaboration with Dr. Jogger in L'Oréal. And so today I would like to talk about the salicylic acid derivative that extends the lifespan of C. elegans by activating autophagy and also mitochondrial UPR. So here is outline of my today's talk. First, I'm going to talk about the introduction and giving you the background that why we started to work on this project. And then I will talk about C. elegans as a model organisms, and then I will explain about aging pathways more in details, and then I'll show you the results in C. elegans in human cells, and at the end we have the conclusions. So just to give you a background that why we started to work on this project, and actually there are records going back to thousands of years showing the beneficial properties of extracts from willow barks. And these extracts are probably one of the most ancient remedies to reduce fever and also ease aches. So then the scientists were curious to find out what are the molecules that are important in these extracts. So then they found that the main active molecules present in these trees are salicylates. So then, of course, they started to investigate more about the salicylates. So it has been shown that salicylates have beneficial activity on different pathways that are implicated in longevity and lifespan. Actually, they increase the lifespan of different model organisms such as yeast, worms, flies, and also mice. So because this work has been done on worm, I'm going just to talk about how it's happening. So it has been shown that salicylates increase the lifespan of worm by activation of TAF16, which is a FOXO transcription factor and is an insulin pathway, and also by activation of AMPK that is important for sensing the level of the energy in the cells. So having these results led us to investigate another derivative of salicylic acid, which is 5-octanol salicylic acid. So in this derivative, we have an octanol group in a metaposition of the acid group. And the interesting part about this derivative is in 2007, it has been shown that it has the lipophilic nature, and also it penetrates slowly in the skin, and it has a skin exfoliating activity. So then we were wondering if there is a positive effect on a worm's lifespan. So talking about worms, first I would like to talk and introduce you C. elegans that we used it for our aging as a model organism for the aging studies. C. elegans is a small worm, it's about 1 millimeter, and it has a short life cycle. It's about 3.5 days. And so as soon as they become adult, they give like 300 progenies. And also it has a lifespan of 2 to 3 weeks, which is make it more practical for aging studies, and it's much quicker. Then it has a transparent body, and it's make it as a convenient model for microscopy studies, and we can see all the details of the body of inside the body of C. elegans. It's easy to maintain in the lab, so we feed them with the bacteria on agar plates. And we have large mutant and strain collections, and if we need to keep them for a long time, we can always freeze them at -80. And also genome sequence is known. So having this derivative that I talked earlier, so we wanted to know if it has a positive effect on C. elegans lifespan and half-span. So for lifespan studies, here we are looking at the lifespan of wild-type worms. So first of all, we synchronize the eggs, and we let them to grow up until day 1. And then from day 1, we start to count them every other day, the dead one and also the alive one. And here we have the wild-type worms exposed to C8SA. And as you can see here in red, they are living around 19% longer than the untreated worms. And also they look healthier for us. So then we study the health span of worms treated with C8SA. And so interestingly, we found that so when worms are exposed to C8SA, they move more intensely. For this experiment, we counted the number of body bends per second, and we found that they move more intensely and also at a later stage of their life. So in another experiment, we also look at protein carbonylation, which is a hallmark of aging. And we could see that when they are exposed with a C8SA, they show less level of protein carbonylation. So by now, we have this derivative that has a positive effect on C. elegans health span and also lifespan. So then the next question is, what are the mechanisms that are involved in the effect of C8SA? So to answer this question, we started to investigate some of the aging pathways. So we know that there are some known aging pathways that by modulation or downregulation of these pathways, we can extend the lifespan in different species. And so they can be like insulin pathway, dietary restriction, or mitochondrial pathway. And also there are some more pathways that here I'm just presenting these 3. So we started to investigate the role of insulin pathway to see if it has any role in the effect of C8SA. So in insulin pathway, we have DAF2 as an insulin receptor and also like downstream genes. And so when this pathway is active, it phosphorylates DAF16, which is a transcription factor. And when it's phosphorylated, so it stays in the cytosol. And when, for any reason, this pathway is not active or worms are under stress, and DAF16 doesn't get phosphorylated. So it goes to the nucleus to regulate the expression of the genes that are important for stress resistance or longevity. So then we wanted to know if C8SA has any effect on DAF16 translocation. Is it in the cytoplasm or nucleus? In order to answer this question, we used worms carrying DAF16 GFP, and as you can see here, so DAF16 is everywhere in the cytosol, and when we put them on heat shock, DAF16 goes to the nucleus, so here are the nucleus, and DAF16 is inside, just to regulate the expression of the genes that are important to be resistant to heat shock. And then when we treated them with C8SA, we couldn't see any changes in the translocation of DAF16. So although we had these results, but also it has been shown that even just a little bit amount of DAF16 in the nucleus can be efficient to regulate the target genes. So we look at one of the target genes of DAF16, which is SO3, and it's superoxide dismutase, and it's important for stress resistance, and we could see the level of expression increases. So to confirm this result, we also did some lifespan studies, and here we used DAF16 mutants. So as you can see here, when we exposed them to C8SA, we couldn't see the beneficial effect of C8SA on lifespan anymore, and this suggested us that C8SA extends lifespan of C. elegans through the insulin signaling pathway. So then in next step, we investigated another aging pathway, which is dietary restriction. So dietary restriction is a reduction of food intake without any malnutrition. And for the first time, Mackay in 1935 showed that the reduction of food intake of rodents increases the lifespan. But it's important to know that until 2007, we didn't know about the molecular mechanisms of this effect. So then we were wondering if C8SA also works in dietary restrictions. So to answer this question, we did some lifespan studies and we applied different methods to do dietary restriction. So here we have the wild-type worms on ad libitum. AL is ad libitum, and it means that when we just grow the bacteria and we feed our worm with a normal amount of food, which is ad libitum. And dietary restriction is when we diluted the bacteria. So the worms have less bacteria, and as you can see that they are living longer. And then the third condition is bacterial deprivation, and it's when worms are adult, we put them on plates without any food. And it's very interesting because C. elegans is one of the rare model organism that can stand the complete bacterial deprivation. So there is no food until the last day, and you can see that they are living much longer. So then when we exposed the worms on C8SA, we couldn't see any effect of C8SA anymore, and that also suggested us that C8SA might work in dietary restriction pathway. So as I said, until 2007, we were not sure about the molecular mechanisms, and then there were some genes and mediators that we found that these genes and mediators are responsible for the effect of dietary restriction response. And so we tested some of them, and today I'm just going to present two of these genes. So the first one is AMPK. As I showed at the beginning, it has been shown that salicylates increase the lifespan of worms by activation of AMPK. And we wanted to know also if this is the same case for our derivative. And we found that when we used the AAK2 mutant, so in AAK2 mutant, we don't have the activation of AMPK anymore. And we could see that the effect of C8SA on lifespan extension is missing here. So this suggested us that C8SA increased the lifespan of worms by activation of AMPK, or in another word, activation of AMPK is important for the effect of C8SA on lifespan. Then in next step, we moved on to TOR signaling and autophagy. To check TOR signaling, we looked at one of the targets of TOR, which is DRR2. It's a eukaryotic translation initiation factor, and we could see that when we exposed the worms to C8SA, we could detect a significant reduction of the expression of these genes. So then this result suggested us that TOR is inhibited when we move the worms to C8SA. So then if TOR is inhibited, so then we were expecting to have more level of autophagy, and actually that was the case. So to do this experiment, we used transgenic worms that they were carrying LGG1 GFP, which is an autophagy readout. And as you can see, there are some cells here that I'm showing them here. So we call them SIM cells. And then we can count the number of the foci that they are the autophagies or accumulation of the LGG1 GFP. And as you can see here, so as a positive control, we have dietary restriction that increases autophagy. And also when we expose the worms to C8SA, we could see that the level of autophagy is increasing. And then for the negative control, we have BEC1 RNA is one of the autophagy genes, and as soon as we don't have it, so the level of autophagy decreases. So talking about BEC1, so, and also we wanted to confirm these results and show that the autophagy genes are important for the effect of C8SA. So then I use another mutant, which is an autophagy mutant, and we could see that we don't see the effect of C8SA anymore. So we can say that C8SA activates various processes usually linked to dietary restriction. So this is the last aging pathway that we also tested. It's mitochondrial pathway. So we know that modulation or downregulation of the subunits of mitochondrial complexes increases lifespan and also triggers mitochondrial UPR, which is unfolded protein response and is important for the survival of the cell. And that also increases lifespan. So in order to see if we have C8SA is functioning through mitochondrial pathway, we use one of the mitochondrial mutant, and we could see that the positive effect of C8SA is not there anymore, and that suggested us that C8SA is also working in mitochondrial pathway. So then we wanted to know if the mitochondrial UPR is important for this effect. And here we have the mutant that are defective in mitochondrial UPR, and we could see that the positive effect of our compound was not there, and we don't see the further longevity extension, and we could conclude that activation of mitochondrial UPR is important for the lifespan extension of effect of C8SA on C. elegans. So by now, we know that C8SA is working in insulin pathway, dietary restriction, and also mitochondrial pathway. And here are the mediators that are important for the effect of C8SA. So we know that it increases lifespan through activation of mitochondrial UPR, autophagy, DAF16 transcription factor, and also by activation of AMPK. So now, what about skin? Because we know that worms, they don't have skin and they just have cuticles. But the good news is when we started to do this project and when we had this nice result on C. elegans, we were hoping to show the conservation of the role of C8SA in higher model organism, or let's say more complicated, such as cells. And actually that was the case. So then we were collaborating with a lab in the University of McGill, and we found that C8SA activity is conserved also in normal human keratinocytes. So first of all, we measured TOR activity, and by measuring the phosphorylation of RPS6, and we could see that treating the cells with 30 macromolar of C8SA decreases the level of phosphorylation. That also confirmed the results from C. elegans. So by having less TOR, we were expecting to have more level of autophagy, and that was the case. And we could see that C8SA induces autophagy in a dose-dependent manner in cells. So also, we showed that AMPK is active in cells when they are treated with our compound, which was the same case in C. elegans, and we did it by measuring the phosphorylation of the PACC. And we see that after 3 days of treatment, the level of phosphorylation of the target is increased. So just to conclude, we can say that C8SA extends C. elegans lifespan and health span, and also it increases longevity of worms by activating autophagy and mitochondrial UPR. And also it increases autophagy in human keratinocytes. And as I showed you before, in 2007, so we found that this derivative has exfoliating activity. And so we can say that also it might have some anti-aging benefits on the skin, because there are some other results that I didn't show you here, but we showed that it's also important for the differentiation and also mitochondrial biogenesis. So if you're interested more in the results, we can always talk, or here is the paper, and that is just published in Aging Cell this September. And at the end, I would like to thank Dr. Aguilanius at ONS, Dr. Jagger at Paris, and also L'Oréal, and also our collaborators in University of McGill. And thank you very much for your attention, and I'll be happy to have any questions.
Thank you very much, Mrs. Shamal Nassab, for this very nice presentation. Is there any question in the
Thank you.
assistance? Microphone.
Merci. Thank you very much. Actually, two questions. One is your derivative. Does this also have the COX-1, COX-2 inhibitory activity of salicylic acid? And if did you look at that? And the second question is, because that might be even upstream, from what you observe. And the second question is, did you compare the potency of that derivative to known TOR inhibitors like rapamycin or the homologous substances?
So for the first question, sorry, no, we didn't check that one. And for the second question, if I was understood very well, if we did it with a TOR? No,
right.
rapamycin?
Right. Since you're inhibiting the TOR pathway, a control that you could have run side by side would be to use, rapamycin or rapalox to see the potency of how good your molecule is in comparison to the to other TOR inhibitors.
Actually, yeah. Here we have rapamycin, and then we can see that even the effect was but also for sure it depends on the concentration. But the effect was more significant by using our derivative.
Beautiful, thank you.
Yeah.
Question, Lionel Breton.
Thank you for your talk, Bernard. So just to answer to your question about COX-1 and COX, Professor Gridari, just to answer to your question, the compound have no effect on any kind of COX activities. That's why it's of interest for L'Oréal Corporate. Just to answer to your question. Bernard, just to go back to the classical fields of C. elegans and all the people working on these fields. So, do you know if, because you are more involved in this topic than we can do,
Yeah.
Do you know if Calico Corporate, which is for anyone the global anti-aging firms created from scratch by Google 2 years ago, do you know if Calico Corporate is working on the same target, for example, and mentions that in classical communication or something like that? Why I ask this kind of question, because I see my colleagues, they are laughing just on my left, is so, because the main expert of the world working on C. elegans is a very close number of people and experts all over the world, and Calico probably is one of the main competitors to propose something in the future to prolong healthy span of human. And the question is this kind of topic of target is of interest for us, or if it's not so diffuse and it's for the moment extremely secret by the company?
It's a very good question, because I would like to know too, because whatever Calico is doing is confidential, and okay, no, it's okay. So we had a postdoc in our lab that moved to Calico, and now it's just like 5 years that he's working there. But whenever we ask him any question, he won't answer it because everything is confidential. But for sure, we know that they work a lot on C. elegans and also in cells, but I think they just work on C. elegans because it's much faster, and most of the genes are conserved in human cells and also in other model organisms. So that's why they started to work on C. elegans. But if they're interested in this one, I have no clue. I can ask my friend if you want. Yeah. I told you, maybe at the beginning you didn't hear it. You're welcome.
Okay. I have a question about the skin part of your talk, sorry. Do you know if the exfoliating action of the C8AC could be linked to the modulation of keratinocyte differentiation by autophagy in the epidermis? It was studied by your collaborators?
As I said, it's done with our collaborators, but we know that there is also changes in the, so there were some markers, like differentiation marker, were also upregulated. And we know that because when AMPK is activated, and because we showed that AMPK is activated, so then it activates the target genes. And it's important also for activation of some of the differentiation genes, which they are there.
Thank you.
Questions there?
Thank you very much for the interesting talk. I have a translational question as well. So at least for your in vitro assays, you use quite high doses of 30 to 100 micromolar. So, and this is quite high doses, speaking from a pharmacological perspective. Can you comment on that?
I mean, you're talking about this experiment, yes?
Yeah, but the doses for rapamycin are 100 nanomolar, and for your compound you have to use 100 micromolar. So the question is, would this be applicable to oral treatment, or would you need such high doses that this is not possible as a treatment?
To be honest, I wouldn't, I don't know this one, yeah. Because, I mean, I was responsible for the works on C. elegans.
Thanks.
But, I mean, in C. elegans, we did a dose dependent, and we found that the best concentration was 100 micromolar. Yes.
No more questions?
I was wondering if there is any effect of your compound on ROS production, and if ROS production has any role in the extension of lifespan and etc.
We didn't check if there is any effect on ROS production, but what we checked is we had less level of protein carbonylation, which showing that we have less level of oxidative stress. So if I answer your questions.
Okay.
Like it can be like indirectly showing that we might have less level of ROS production.
Last question, after we have to continue.
It's a comment, it's not a question. Just to come back to the dose with the rapamycin. Rapamycin is a direct inhibitor of mTOR, and the effect of this compound is indirect. So I think that would explain that you have you need much higher dose because it's an indirect effect of AMPK activation and so on. That was just a comment to answer this question. Thanks.
Okay, thank you. Thank you very much, Mr. Shamal Nassab. Now I call Mrs. Regina Weinmüllner to speak about organotypic human skin equivalents. Mrs. Weinmüllner is biotechnologist, PhD student. She is in University of Natural Resource and Life Science, Vienna, Austria.
Thanks for the invitation to speak here. Topic of my talk is organotypic human skin equivalents, a novel model to study the influence of aging and environmental pollution on skin development. Just as a disclaimer, my work was partly financed by the Christian Doppler Forschungsgesellschaft, which bridges research between university and an industry partner. Our industry partner is Chanel Research and Biotechnology. Why am I interested in tissue-engineered human skin equivalents? As a scientist, I want to stress the principles of 3R, meaning I try to replace, reduce, or refine animal experiments. Tissue-engineered human skin equivalents are substitutes to mimic the human skin. Depending on what cells you seed in, you have different models with increasing complexity. They are also used as skin grafts in patients. As you've already heard from my colleagues before, cells have a limited lifespan and enter then a state called cellular senescence. Hallmarks of senescence are the irreversible growth arrest, the resistance to apoptotic death, the senescence-associated secretory phenotype, shortly SASP, and they are positive for senescence-associated beta-galactosidase. The senescence is normally induced either via telomere shortening, DNA damage, or activation of oncogenes. Why am I now interested in the effects of senescent cells on skin aging? Senescent cells accumulate in the aged skin, and the removal of senescent cells prolongs the healthy lifespan in various mouse models. As you can see in the cartoon below, in aged skin, the fibroblast, let's see, it's here, this is the fibroblast, changes its state and starts to produce SASP, mainly with matrix metalloproteinases and inflammatory cytokines, therefore remodeling the ECM. I will tell you today about two different skin equivalent model systems. On one hand, we used senescent fibroblasts and studied their effects on skin aging. On the other hand, we immortalized keratinocytes and used them in a chronic environmental pollution model. But first, we start with the aged skin equivalent model. How do we actually build skin equivalents with senescent cells? We start either with young fibroblasts or stress-induced premature senescent fibroblasts and put them in a collagen matrix. Then we seed keratinocytes on top and lift the whole thing to the air-liquid interface, where the keratinocytes start to differentiate by their own and form the epidermis. After 7 days, I can harvest the more or like calamari-like structures and can then stain and analyze them. First, we wanted to make sure that we really are working with senescent cells. Therefore, we did on one hand an ESA beta-gal staining. And as you can see here in the graph, we had 70% ESA beta-gal positive cells in our SIP cells. As a second assay, we implemented BODU assay checking for cellular proliferation, while our young proliferating cells were 60.4% BODU positive. Our senescent cells were only 7.4% BRDU positive left. The next question we asked is, are our senescent fibroblasts actually surviving the whole process of being seeded into a skin equivalent? It's fair to say, well, they are already stressed, and then you put them to more stress, so are they just dying in the skin equivalent, or what's happening? Therefore, we isolated the cells on different days from the skin equivalent and counted them. Additionally, we reseeded them to make sure that we are counting viable cells and to make sure that we have viable cells. As you can see here, we don't lose any cells over the duration of the whole experiment. Next, we were interested if our senescent cells actually have any influence on the formation of the epidermis. Therefore, we added increasing amounts of senescent fibroblasts into the skin equivalent and decided to use epidermal thickness, which is one of the hallmarks of epidermal thickness as a marker. Epidermal thickness is one of the hallmarks of human skin aging, and seemed therefore suitable for us. As you can see here in the pictures and already have seen in Johannes' talk, between 0% senescent cells and 100% senescent cells, you see a clear reduction in epidermal thickness. We also made sure that this effect is not because the collagen contraction in aged skin equivalents is not working properly, by just testing it here. One could argue now, well, you have 100% senescent cells in the mix, but that's not a natural setting that you ever encounter in a human being. Yeah, that's right, but in a human being, you have normally months or years where the senescent cells can influence their surrounding, while in my experiment, you have only 7 days where the cells can do their job. Therefore, we said, well, okay, we take what we have, we do all further experiments with 100% senescent cells and stained for epidermal differentiation markers. We stained for keratin 10, which is an early differentiation marker, and saw a reduction in keratin 10 positive layers. And we stained for filagreen, which is a late differentiation marker, and we're surprised to see that we even have some spots here where nearly no filagreen staining is left, hinting that we have troubles with terminal differentiation. Our next question was, is the problem with terminal differentiation reflected in barrier integrity? Therefore, we implemented a so-called barrier assay. We put the drop of biotin on top and just let it soak in. And if the barrier is intact, you get just the outermost layer stained. And if the barrier is defect, you get a whole green skin equivalent. As you can see here, we saw a trend toward more barrier defect skin equivalent when we built aged skin equivalents. What we encountered while we were doing the experiment was that the surface properties of our aged skin equivalents seemed to have changed. To analyze this effect in more detail, we implemented a contact angle measurement method that was available at our university. You can see it here. It's basically just putting a drop of water on the skin equivalent and filming the movement of the water on the surface. Analysis of this data revealed that actually we have changed surface properties. The drop spreads quicker, and Also, the data varied more when we have aged skin equivalents. Next, we wanted to know if the changes between young and aged skin equivalents could also be found in the supernatants surrounding our skin equivalents. Therefore, we used a technique called Raman spectroscopy, which is a label-free method to analyze the chemical composition of cells, substances, or supernatants. In the beginning, we were not very lucky. You see it here. Because what you expect normally is a very nice spectra where you then can assign peaks. So we improved our method by adding a process called SIRS, where you bind the components of the supernatant to gold particles. This strongly increased the quality of our spectra. And with a little bit of magic from principal component analysis, we were first able to identify senescent cells versus quiescent cells in the Raman spectra and can separate them via the Raman spectra. And can also do that with skin equivalents in 3D. With supernatants from skin equivalents in 3D. Yeah, we expected that most of the changes in the composition of the media would be from SASP. I mean, we know we have senescent cells in there, senescent cells produce the SASP, so yeah, it was quite obvious. Well, as the title suggests, it's not always obvious in science. I've put here the timeline of my experiment again so that you have a better overlook. On day 1, I see the fibroblasts. On day 2, I see the keratinocytes. On day 3, it's recovery. And on day 4 starts the actual differentiation process. And if we now just look on day 1, here and here, we see what we expect. Skin equivalents built with senescent fibroblasts have more SASP factors than their young counterpart. But as soon as we put keratinocytes on top of the skin equivalent, or here, the whole thing changes. We have a few ideas what's happening in there, and are planning more experiments to answer that in detail. But at the moment, you have to live with those data. So, taken together, senescent cells in the skin equivalents importantly survive the experimental setup. They reduce the epidermal thickness. They impair differentiation. They have a negative effect on barrier formation. They modify the surface properties of differentiated skin equivalents. They change the chemical composition of the supernatant, and they dampen the secretion of inflammatory cytokines in the initial phase of the skin equivalent formation. As promised, I have also a second model here with me. We actually wanted to improve a skin equivalent model to study chronic exposure and used arsenic as a model pollutant. I have no idea how many of you are familiar with arsenic as a chronic pollutant. It's one of the most common environmental toxins. It's of high abundance in the earth crust and leads normally to groundwater contamination in some areas. The symptoms of chronic toxicity usually manifest first in the skin. and lead to hyperpigmentation, hyperkeratosis, Bowen's disease, that is a carcinoma in situ, squamous cell carcinoma, and basal cell carcinoma. And more importantly, since mouse models don't show skin symptoms of chronic arsenic intake, there are no animals available to study chronic effects. We've heard before keratinocytes have a limited lifespan, so chronic exposure over months is not possible. To date, Hackard cells are used instead. They are spontaneously a spontaneously transformed keratinocyte cell line, but they have an incomplete differentiation pattern and lack of several markers that primary keratinocytes have. Therefore, we developed an immortalized keratinocyte cell line called NHEC S4TERT35 in our lab in cooperation with the spin-off Eversight GmbH and the Medical University of Vienna. Compared to the state-of-the-art Hackard, here, our cells showed a thicker epidermis, more keratin 10 marker, which is an early differentiation marker compared to here, and beginning of late differentiation marker filler green here in comparison to the Hackard cells here. Our treatment scheme then was to treat the cells during serial passaging over 6 months biweekly with arsenic and characterize the cells then in 2D and 3D to study the chronic effects. In 2D, the chronic exposure with arsenic leads to an accumulation of the cells in G2. You see it here, 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 and here. And more importantly, the cells also didn't develop any arsenic resistance over the 6 months. It's seen here. Additionally, we saw an increased rate of ROS level over the whole time of the experiment. When we used these chronic exposed cells to build skin equivalents, we were surprised to see that, especially here and here, the skin equivalents were no longer topped with a nice stratum corneum here, and a bit here. And when we stained for the proliferation marker key 67, we saw that the proliferating cells were no longer confined to the stratum basale, but started to migrate to the whole skin equivalent up to the top of the epidermal layers. This may be the first step of Bowen's disease. At least it looks similar in patients. Another thing that we encountered was that the cells started to lose keratin N as a differentiation marker. You see it here, which led us to the question, what is actually happening to the cells? We speculated since the cells were growing, and we also saw that they behaved differently in the cell culture. They started to lose their cell-cell contacts and become a little bit more spindle-shaped. We speculated that perhaps they have underwent a partial EMT. A partial EMT means that the cells start to lose cell-cell contact, and is a first step towards EMT, where then EMT markers are upregulated and the cells switch from the epithelial to a more mesenchymal type of cell, where they then can invade the surrounding tissue or the blood vessels. In line with our theory, we saw that the cells started to lose expression of keratin 14, which is one of the keratinocyte markers. And they lose the cell-cell contact, here stained via ZO1. They also could form spheroids, meaning that they don't need, they no longer need a base where they can grow. So they grow now in suspension. And they lost the E-caterin expression and gained the EMT markers TCF8 and SLUG. So in conclusion, our immortalized NAC-S4-35 resembled closer the differentiation pattern of primary keratinocytes than the state-of-the-art HACAD cells. They can be used for chronic toxicity studies at least up to 6 months. The chronically arsenic-treated cells show first signs of in vivo arsenicosis when we use them for 3D experiments and revealed an arsenicosis-associated EMT. Taken together, both models that I presented you today here offer enhanced tools to study basic research questions, either in the field of aging as well as in environmental pollution research. And my last slide, I would first and all thank my boss, Johannes Grillari, for listening to his technician when she told him, well, let's do a PhD. I'm here anyways. I thank all group members who always help me with my experiments and talk with me about it. Thank all the cooperation partners, even more than I could fit onto this page here. And special thanks to Chanel Research and Technology for funding my research. Thank you very much, and thank you for your attention.
Thank you, Mrs. Weinmüller, for this good presentation. And now if you have any question. Martin.
Thank you very much for your presentation. I have a question about the model with the senescent fibroblast. How do you explain the reduction of epidermal thickness? What is the mechanism that is modified in this model leading to this reduction of thickness? Did you analyze some proliferation steps, stratification?
Yeah, we are also quite a bit puzzled here. I mean, the first idea was, well, the skin equivalents just don't contract as much, so you then have a reduced epidermis, but that was not the case. I would speculate it's the factors that the senescent cells secrete that are somehow influencing how the keratinocytes on the one hand proliferate, so that they can't proliferate as quickly as their young counterpart, therefore leading to less proliferation, but who knows. We've also looked for apoptosis, but at least in the last stages of the skin equivalent, where I can actually stain for apoptosis, we don't see much. So if there's apoptosis of the keratinocytes because there are senescent cells in there, it has to happen at the beginning where I can't visualize it. Other than that, yeah.
Question, Lionel Breton.
Yes, thank you for your talk. Really nice model. But so, in the same field, like Jerome's question, so to recreate senescent mammalian epithelia through fibroblast disorder, did you or do you have a look on any kind of relationship between the two partners, and which kind of compound, which kind of, I don't know, growth factor, any kind of mediator could be the link between fibroblast and global epithelium in your model? Do you have a look on this field?
So far, we only tested 4 SASP factors, GM-CSF, IL-6, IL-8, and IL-1 alpha, I think. But we will have a deeper look into it and see if we can figure out what's going on there, actually.
Because your hypothesis is it could be only inflammatory factors that could create this model?
I'm not sure. Fibroblasts. It's a good question. Let me think about it and I come back with you with the answer, okay?
I have 1 day and a half to get your answer. Thank you so much.
Thank you. Thank you. Another question?
To go further with the same question, it is established that the SASP can induce senescence in the neighboring cells. So do you look whether there is some induction of senescence in the epidermis of the reconstituted skin with senescent fibroblasts?
No, I haven't stained for senescence. I've stained for P16 in the whole skin equivalence, and the cells in the epidermis were at least not positive for P16 that I've stained. We have plans to implant isobeta-gal staining for the skin equivalence. That's not as easy as it sounds. There are some technical issues there. But I will look in the epidermis if I see there are some senescent cells too.
Thank you. Another question? So, thank you for these very nice sessions. Now I think we can go directly. The next session is 2 o'clock.